Welcome back friends in this video tutorial of Anita Bioconcept, and today I am going to discuss about the lectin pathway of the complement system. In my previous video lectures I had discussed about the classical pathway of the complement system as well as alternative pathway of the complement system. So there are all total 3 pathways: alternative pathway, classical pathway and lectin pathway. So today our topic is lectin pathway. So let's start with the lectin pathway. So in case of lectin pathway, how does it originate? So we will see that how does the lectin pathway originates. Lectin pathway originates with host protein binding microbial surfaces. Okay, so this lectin pathway, this is the lectin pathway, this is classical pathway, this is alternative pathway, this is alternative pathway, this is classical pathway, and this is the lectin pathway. So lectin pathway originates with the host protein binding microbial surfaces. Host protein binding microbial surfaces. Okay, so what are lectins? The first question arises, what are lectins? Lectins are proteins that recognizes and binds to the specific carbohydrate residues, especially mannose. Okay, so this is one of the reason that this pathway, this lectin pathway is also known as MB lectin pathway, means mannose binding lectin pathway or mannan binding lectin pathway. So this pathway is also known as MB lectin pathway. Okay, so like alternative pathway, this lectin pathway do not depend upon the antibody for its activation. Okay, the mechanism is more similar, but the mechanism is more similar to classical pathway. What we see that this lectin pathway do not require antibody, just like the alternative pathway, but its all mechanisms are almost similar to the classical pathway. Now the question arises, why? Because after initiation it proceeds through C2 and C4. After its initial initiation or after its activation, it proceeds through C2 and C4 to produce the activated proteins of the complement system, and so it is more likely similar to the classical pathway. Okay. Now, how lectin pathway is activated? Okay, so it is activated by the binding of MBL, means mannose binding lectin to mannose residues on the glycoproteins or carbohydrates. So this lectin pathway begins by the binding of mannose binding lectins to the mannose residues on glycoproteins or carbohydrates on the surfaces of microbial cells such as Salmonella, Neisseria strains, some fungi, some viruses such as HIV-1. Okay, and then after, okay, now about MBL, mannose binding lectins. It is a member of collectin family. It is mannose binding lectins are the members of, these are the members of collectin family. Okay, MBL is an acute phase protein and its concentration increases during inflammatory response. Now what is its function? Its function is similar to C1q. The function of MBL is similar to the C1q. After MBL binds to the carbohydrate residues on the surfaces of cell, microbial cell wall or microbial cells or pathogens, this MBL associated serine protease, that is MASP, MBL associated serine protease MASP1 and MASP2, MASP1 and MASP2 binds to the MBL, and the active complex formed by this association causes cleavage and activation of this C2 and C4 complement protein. The MASP1 and MASP2 protein are structurally similar to the C1R and C1S, and their activities are almost similar. So then after it activates the C2 as well as C4 components, so a C5 convertase is formed after that, and there is no need of antibody binding. So after C5 convertase formation, and then after C5b peptide fragment does binds to C6, C7, C8, C9, and after that the formation of MAC will be taking place, and it will lead to the lysis of cell. So this is the lectin pathway. So we have seen that all three pathways does lead to C3 convertase, C5 convertase as well as C5b, and then after C5b leads to formation of MAC and it will lyse the cell. So this is all about the three pathways of complement system, that is classical, alternative as well as lectin. So today I have discussed about the lectin pathway. Once again I am reviewing that. Okay, so first of all, what happens, the lectin pathway do not depend upon the antibody for its activation. The mechanism is more similar to classical pathway. Why? Because because after initiation it proceeds through C2 and C4 to produce the activated proteins of the complement system. Now what happens? Okay, now how the lectin pathway is activated? It is activated by the binding of this MBL to mannose residues on the glycoproteins or carbohydrates on the surface of microbial cells such as bacteria and viruses such as HIV-1 and some fungi also. MBL is a member of lectin family, so it is an acute phase protein and its concentration increases during inflammatory response. Now what is the function of this MBL, mannose binding lectin? When mannose binding lectin or after MBL binds to the carbohydrate residues on the surface of these microbial cells or pathogens, MBL associated serine protease, that is MASP. So MBL associated serine protease 1 and MBL associated serine protease 2 binds to the MBL. The active complex formed by this association causes cleavage and activation of the C2 and C4 complement proteins, and finally the MASP1 and MASP2 are structurally similar to the C1R and C1S and their activities are almost similar. So then after its activation, the C2 and C4 components, then after it activates the C2 and C4 complement proteins, the C5 convertase enzyme is formed which generates the C5b peptide fragment, and after that finally with the series of cascade of the reactions, then after C5b does lead to the formation of membrane attack complex by binding to the C6, C7, C8 and C9, and finally the MAC formation will take place, that is membrane attack complex, and it will be responsible to lyse the cell. So this is about the lectin pathway of the complement system.